Good afternoon, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and it's my distinct pleasure to welcome you to this last in our series of the 2024-2025 Foundations of Biomedical Data Science Seminar Series. This unique lecture series this year has been brought to you through a generous support of the Integrated Translational Health Institute of Virginia, the University of Virginia's College of Arts and Sciences, the UVA School of Data Science, grants from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across the diverse fields of biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we have enjoyed presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic-related methods, along with the wide variety of data analytic opportunities they present in data science, deep learning, AI, and spatial modeling. We are particularly excited about this year's Biomedical Data Science Innovation Lab program, where junior faculty, biomedical researchers, and computational experts will develop novel joint projects on the topic of quantitative approaches and applications for spatial multi-omics data types and their use towards guiding personalized medicine solutions. Participants selected for our special program utilize the content from lectures such as these as essential material in our culminating in-person manuscript and grant development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. For today's lecture, I am delighted to welcome our speaker, Dr. Shannon Hughes from the National Cancer Institute. Dr. Hughes is the Deputy Director of the Division of Cancer Biology at the National Cancer Institute, where she helps to oversee the NCI's foundational cancer biology portfolio. She earned a bachelor's in chemical engineering from Iowa State University, and following her undergraduate studies, she spent 3 years working as a process engineer, working in the field of drug delivery at the 3M Corporation. Industry research motivated her PhD in biomedical engineering and a postdoctoral research opportunity focused on breast cancer metastasis. She earned her PhD from Washington University in St. Louis in biomedical engineering, and she was a lead technical instructor in biomedical engineering at MIT. And at NCI, she also promotes emerging scientific areas and a diverse cancer workforce to advance progress in cancer biology. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Shannon's lecture today is entitled Resources at the National Cancer Institute to Support Spatial Biology. Cancer is a complex disease driven by multiple molecular, cellular, and tissue-level interactions, and the application of new and emerging technologies, especially those focused on understanding the temporal and spatial relationships of cancer, is imperative for the discovery of new prevention and treatment modalities. The NCI supports several funding opportunities which facilitate development and application of experimental and analytical methodologies, and Dr. Hughes' lecture today will provide examples of the current technological programs and funding initiatives, as well as tips for finding and applying for NIH funding. As always, we are streaming this lecture live and for recording via YouTube. If you're watching on YouTube, thank you for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have by the chat feature in their YouTube sessions. I'll be synthesizing those questions and asking them on your behalf during the last 10 minutes or so of the lecture. And with that, thank you so much, Shannon. You are our 152nd all-time seminar speaker, so thank you so much. We are really looking forward to your presentation.
Thank you, Jack, for that super kind both invitation and introduction. And I'm in a relatively suboptimal location for giving this talk, so please forgive the background noise, and hopefully everyone will be able to hear me. I'm going to spend the time today talking about many of the things that we support in the Division of Cancer Biology. As Jack already told you, I'm a federal government employee, and because of that, I don't have any financial relationships to disclose. So my position at the NCI as the Deputy Director of the Division of Cancer Biology allows me to see a very broad range of cancer biology science. And today I'm going to provide you a bit of a tour through several NCI-supported programs where investigators are asking interesting and important questions that really rely or need a spatial biology component to address. I'll do my best to describe how adding that spatial component has contributed to our knowledge base around cancer biology, how it's pushing things towards the clinic that otherwise maybe wouldn't be going there, and the many people that are working to make those spatial biology data sets and computational tools available to the community. So previously in this seminar series, Dr. Jill Barnholt-Sloan from the NCI presented a large number of large-scale, high-value genomics and clinical imaging data sets. One of the first endeavors that the NIH took was a collaboration between the National Cancer Institute and the Institute of the NIH that supports genomics research. And that collaboration was the creation of the Cancer Genome Atlas. The Cancer Genome Atlas, or the TCGA, over the course of its 15-year lifespan, which wrapped up just in the early 2020s, analyzed around 11,000 patient samples across 33 tumors. Almost all of those samples were collected from patients that were pre-treatment, and many of those samples were early in the disease. And those decisions were made very purposefully to minimize confounding variables. The TCGA effort is arguably the most successful large-scale genomics effort that has been funded by the NCI, although you also heard from Sean Hanlon about the Human Tumor Atlas Network earlier in this seminar series, which I think is also generating a lot of really interesting multimodal data. But the TCGA really launched our ability to identify actionable genetic alterations that can be treated by any number of therapy options. Now you can find the TCGA data set on the NCI Genomics Data Commons. All of the TCGA data is available through the NCI Genomic Data Commons, or GDC, along with a number of other data sets. The total number of patient samples that have been analyzed where data is available to explore and create new hypotheses is about 45,000 on the NCI Genomic Data Commons. An important aspect of the GDC is that all of the data has been harmonized using a validated set of computational tools, including consensus somatic mutation calling, tools to analyze transcriptional signatures across these tumors. And importantly, I think one of the high-value data sets that's new within the NCI GDC are about 9,000 whole genomes from the TCGA data set. All of these data can be analyzed with a number of different tools on the GDC, and the GDC specifically not only provides access to raw data once someone is able to apply for controlled access, data access through dbGaP, but also derived data that people can explore using a number of computational tools. So the GDC's one of the goals of the GDC was to provide a place where people without a computational biology background can go and glean insight, combine their data with data from other data sets like the TCGA, and try to, again, derive these hypotheses that can lead to personalized medicine. There are, of course, a number of other large cancer data sets that have been stood up over the course of the last 5 to 10 years. For example, one of those examples is Project Genie, which is sponsored by the American Association for Cancer Research. And you can see from this slide that they have almost 230,000 patient samples that are available for browsing through a different portal. The Genie portal is hosted by a team that's behind the CBio portal, which is the most, I think, one of the best ways to analyze the cancer data. And the CBio portal team is supported by a number of agencies, along with the NCI through the NCI Informatics Technology for Cancer Research program, which I'm going to talk a little bit more about later. So all of these data sets, either the ones that are within the TCGA data set, along with things, data that is presented by the Genie project, are on bulk tumor samples, which means that we're looking at an average of the tumor at any given time, and perhaps even within a sample that's taken from a tumor, we're looking just at one part of it. So we're perhaps not getting the totality of the cancer heterogeneity by looking at these large-scale genomics assays. But one thing that large-scale genomics have done is they've provided a route to clinical treatment. And really one of the things that's most important about that is the harmonization and the careful curation of these large cancer data sets, which is both resource intensive. For example, it takes several months for thousands of whole genome sequences to be processed in a uniform manner and made available on the NCI GDC. But these large data sets have already demonstrated patient impact. There are a number of examples of new ways to stratify patients based on a somatic mutation signature or based on a transcriptional program that help clinicians now make decisions on how to treat those patients. And this standardized sample processing, validated molecular analysis, characterization assays, along with the harmonized data processing, allows folks to compare across tumor types and across data sets instead of just within a single study or within a single tumor type. And these things are important in that, for example, beginning in, or it's published around 2018, the TCGA team did a pan-cancer analysis. And that pan-cancer analysis looked for somatic mutations, transcriptional phenotypes, methylation signatures that were common across cancers, not just within one tumor. And that has really transformed the field in that it allows clinical trials to be stood up where a patient, independent of what type of cancer they have, can be treated with a drug that's targeting that mutation, these basket trials. So that's really changed clinical practice. In addition, as these large cancer data sets continue to grow, and the number of therapies that are developed in order to target the somatic mutations or other alterations in the genome grow, we also see that the number of patients that are eligible for targeted treatment is growing. This bar plot here on the right-hand side of the slide is another data set from the MSK, the Memorial Sloan Kettering Cancer Center. They are sequencing the samples that come from almost all of the cancer patients that are enrolled in that center. This impact data set has been mined for many useful insights, including understanding the genomics behind different metastatic sites. But one of the more recent papers looked at the number of patients who are now able to enroll in a targeted clinical trial. And what they see, if you concentrate on this kind of light peach color here, is that the percentage of patients with no actionable driver mutation, now there's no drug out there to target this, has shrunk by almost 50% between 2017 and 2022. So these cancer genomics activities are really driving differences in the way that we treat patients. I want to point out here on the left-hand side, these little Petri dishes. So this pan-cancer analysis that was done by the TCGA team, it ended with a number of consensus mutations across tumor types that maybe had not been described before. And so one step in understanding how mutations give rise to things like cancer is these validation steps. And in that case, the validation was a functional one in which the somatic mutations that were discovered through the TCGA pan-cancer analysis were introduced into cell lines, and those cell lines were assayed to see if they gained cancer phenotypes like increase in proliferation or migratory behaviors. And these types of functional validations have given rise to a field called functional genomics. And that functional genomics field has been supported by the NCI through the Cancer Target Discovery and Development Network, which was started in 2010, so about 15 years ago now, by the late Daniela Gerhardt. And the functional genomics, the point of this network and folks who are in the functional genomics field, again, is to move the insights that we're getting from these large-scale data collection and analysis exercises towards actionable things like new drugs that can impact the patients and patient caregivers. In the last 15 years, the CTD Squared program has really defined the field of functional genomics and provided a number of standards for the rest of the participants in the field to follow. For example, this figure is one from a paper that the team published in 2016, talking about different tiers of evidence that are required for translation. So depending on the goal, whether it be drug target identification, development of prognostic biomarkers, or the validation of a drug against a computationally defined target, there are different steps towards validation. So these principles have really stood the test of time, where you first have a hit that's derived from a functional genomic screen. In many cases, these functional genomic screens are computational based, but they can also be traditional screens like siRNA screens or CRISPR screens. Once you identify a gene that causes a cancer phenotype, you need to both test it in vitro and then move on to testing it in vivo. And, these kind of tiers of evidence have been valued as you move towards FDA approval for different targets and tests within the cancer community. But the FDA has just recently said that they may no longer need to have a in vivo validation in a model system like a mouse. And furthermore, almost all of the studies in functional genomics up until the last few years have been focused exclusively on the tumor cell. And we know that the tumor cell isn't the only cell within the tumor microenvironment. So what are the other things that we're missing in the field of functional genomics to truly take us from cancer genetics or cancer genomics to the clinic? One of those things is an understanding of how the tumor microenvironment evolves over time. Because, like I mentioned, we know that tumors are not just made up of tumor cells. So most cancer therapies, with the, I think, big and important exception of immunotherapies, were developed through in vitro assays that really only involve tumor cells. And to be quite frank, that has been successful. There are a number of tumor treatments or cancer therapies that work very well by only considering the cancer cell. However, we know that this is an oversimplification of the cancer ecosystem. And in fact, this overly simplified diagram is a good illustration of what can happen under treatment. The number of cell populations may change. There's good evidence that many of our targeted therapies in cancer target not just the tumor cell, but the immune cells within the microenvironment or the other stromal components within the microenvironment. So just treating with a drug will significantly change the types of cells that tumor cells are interacting with and the types of communication within the tumor microenvironment. So the latest generation of the Cancer Target Discovery Network, investigators have tried to tackle this through a variety of different approaches, such as creating more complex screening systems, doing large-scale drug screens or CRISPR or siRNA knockdown screens and organoids or more complex systems. But one team led by Brian Brown and Miriam Murad at Mount Sinai have taken a slightly different approach, which I think is really interesting and is generating some really great data that calls for really advanced computational analysis. Okay. So this approach is called PerturbMap. And what PerturbMap does is introduces small protein sequences that can be recognized through multiple rounds of immunofluorescence imaging. And this bar-coded nature of the tags, I'm not sure if you can see my mouse, but if you can. So each one of these PerturbMap constructs has both a pro code, which is a small series of proteins, a small series of peptides that is tagged to a CRISPR guide RNA, which functions to knock down genes, right? So now what happens with the PerturbMap is when the cells are infected with this system and injected into a mouse or a system of your choice, tumors can be allowed to form. And I'll give you an example of that in just a second. And after tumors form, you can look at the tissue to determine where the specific guide RNAs ended up within the organ. Okay, I'm going to do this in essentially kind of real-time monitoring of which genes are important in tumor formation in different parts of the body. Okay. So one of the first examples of this, the team was looking both at lung cancer as well as breast cancer. And the examples I'm going to show you are in lung cancer. So here they created a CRISPR library of genes that are important in cancer initiation and progression, tagged each one of those CRISPR guide RNA constructs with a pro code, so this small series of peptides that can be recognized through immunofluorescence, transduce them into a mouse and let them grow. The tumors form for about 4 weeks. After 4 weeks, they sacrifice the mice to look at how different knockdowns of genes that we know are important in cancer impact tumor growth and where they do that within the tissue, which is the part that we were not able to do before. So here's an example of a perturb map in action. So this is a commonly used KRAS mutant lung cancer cell line that was infected with the perturb map system. Again, the pro code peptides that allow us to figure out where the CRISPR guide RNAs are going. After 4 weeks, we have tumors within a lung. This is a histological section of a lung on the left-hand side here. Next to it is a pseudocolored map of where the different clones of the tumor grew. And each one of those different clones has a different CRISPR guide RNA infected into it, meaning that there's a different gene knockdown in each one of those clones. Why is this interesting? It's interesting because before these types of assays, we would have had to dissociate the tissue and do some sequencing to understand which guide RNAs were enriched or depleted. So which ones promoted tumor growth or don't promote tumor growth, but we would have had no idea where they were within the tissue. So decoding this beautiful immunofluorescence image, we can see that there are different genes that within this KRAS mutant lung cancer context drive the loss of those genes, drive tumor formation, such as SOX1, STAT1, IRF9, etc. Many interesting things can be derived from this, including the size of the clone, how dominant is it, the diffusivity of the clone, are the cells being, are they more motile, for example, within the tissue, and the interaction between the different clones that have different gene knockouts. So are there clones that are more likely to be near each other or not be near each other within the tumor? Excitingly, you can couple the perturb map system with spatial genomics or spatial transcriptomics. So here on the very left-hand side of the slide, these KP lesions, those are the ones that have a CRISPR guide infected in them. You can see exactly the different clones in the middle. So each of the red areas is the enrichment of one of the CRISPR guide RNAs, the knockdown of a gene, and its diffusivity around where their clone was, so how large those clones were. Now, on an adjacent sample of this tissue, you can perform spatial transcriptomics. And in this particular example, the Brown Lab performed 10x Visium spatial transcriptomics. And what you're looking at here is the expression of the TIMP1 gene on the right. And you can see that the overexpression of the TIMP1 gene only corresponds to a couple of the different clones within the tumor. So now we can map exactly how the knockdown of a specific gene spatially within the tumor results in a phenotype change. So really mapping genotype to phenotype, which is kind of the holy grail here. Just as another example, this is a gene that encodes for a surfactant that's important in lung function. And you can actually see that it has the opposite localization of an upregulation of a protease gene TIMP1. So there's all sorts of information that can be gleaned from this type of assay. And it brings a new meaning to the term functional genomics, because now not only do we can we correlate the knockdown of a certain gene with a gene expression program, we can also correlate it in vivo in a spatially understandable manner. So this team partnered with a team at Harvard to also to employ the perturb map system in the study of ovarian cancer. And by perturbing many genes that were coding for proteins that are secreted with regards, secreted proteins are the way that cells communicate within the tumor microenvironment. So knocking down different secreted proteins gives you an idea how the different cell types are interacting with the tumor. And what they found in this study is that knockdown of IL-4 actually made these tumors more sensitive to immunotherapy. Traditionally, ovarian cancer is not very sensitive to immunotherapies, and it's not been successful in the clinic. And through the employment of the perturbed map system, this team found actually that removing IL-4 made immunotherapy much more successful because it completely changed the immune ecosystem within that area of the tumor. So knowing that allows the team to now suggest that a combination therapy of an IL-4 inhibitor along with immunotherapies might make ovarian cancer patients good candidates actually for immunotherapy. Okay, so oftentimes we don't have human samples that we can use in that way to analyze tissue. And one way to continue to do functional genomics in a human relevant context is by using human models such as organoids or specific cell lines that we know the clinical data and additional information from. And another initiative that was partnered with the TCGA and the CTD² program, also out of the NCI Office of Cancer Genomics, has been the NCI Human Cancer Models Initiative. And the goal of the HCMI program was to develop as many human-derived cancer models as we possibly could over a period of time and make them available for the scientific community as a community resource. These models, which are mostly organoid models and cell lines, along with normal benchmark tissue, are derived from a range of cancers, and they have extensive clinical and molecular characterization data associated with them. Additionally, all of the models, along with the primary tumor and normal tissue from the patients, were characterized using the same pipelines as the TCGA data. So all of this creates comparable data sets and data sets that are available through the NCI Genomic Data Commons. You can search on the HCMI data portal to try to find human cancer models that are relevant to the types of questions that you would like to ask. And right now, there are just over 300 HCMI models that are available. Once you find the models for the tumor type, or like we talked about before, the mutation or genomic aberration that you're interested in, you can drill down into those models. You can see that important patient clinical information, such as the previous lines of therapy that the patient saw before donating their tissue. You can also see the available molecular characterizations that have been done. So for example, this particular model has whole genome sequencing, whole exome sequencing, RNA-seq, and DNA methylation data available for it. You can take a gander at what the model looks like, either a cell line or organoid model. And importantly, you can find the sequencing files for these models that are available through the Genomic Data Commons. And you can purchase these models through ATCC. And the ATCC price for academic and nonprofit organizations is significantly lower than the list price on the website. So make sure that you inquire about these. So right now there are about 300 models available. ATCC has about 700 models that were derived from the HCMI project. And if there are models, if you're looking through the clinical data and there's a model that you really need to have to test a hypothesis, you can also ask ATCC to expand that model for you and send it to you. So we hope that it's a really good resource, and we want a lot of people to use it. Okay, so a burning question, I think, in spatial biology is how much of what we see in vivo in a patient can be recreated in some of these model systems like the ones that HCMI has made available to the community. And understanding how systems evolve both in vivo and in vitro and creating mathematical and computational models in order to predict that evolution is really one of the goals of the NCI Cancer Systems Biology Consortium. You know, although it can seem like second nature now to consider complex diseases like cancer from a systems level view, it really wasn't that long ago when we were really just in our labs studying our favorite gene or favorite protein. So one of the groups that's funded by the Cancer Systems Biology Consortium really wanted to start to ask the question, how can you compare the model systems that we're using, such as organoids, or in this paper, assembloids, which are really purposeful assemblies of human cells that are put together, to what we see in patients. So, it's quite difficult sometimes to determine if a spatial relationship is maintained, because tumors especially are really complex systems. And many investigators, as you probably all know, are finding lots of different representations of tumor neighborhoods or ways to quantify co-localization. In fact, I don't think I stuck the citation in, but there was just a really interesting paper released on bioRxiv in the last week that did a comparison between the ways that people are defining neighborhoods within tissue structures, and they're not all matching, which isn't a huge surprise. But one way that a team funded by the Systems Biology Consortium has decided to address that is by systematically looking at assembloids made from human lung cancer cells to human lung cancer tissues that are intact, to see how many of the co-localizations, so how many cells that are next to each other, how that co-localization is maintained between organoids and the tumor tissue itself. This is work from Silvia Plavridis' lab at Stanford University. And they're, like I said, using assembloid cultures of both tumor cells and fibroblasts, and asking how do the relationships between the tumor cells and fibroblasts in vitro, are they reflected in in vivo context? So they use a technique called Codex, which was developed in Stanford and was also developed using some resources from the NCI that I'll talk about a little bit later. This is a multiplex protein immunofluorescence assay where sections are stained with up to 45 to 50 different markers important in whatever the biology that you're studying. They take the data from these Codex assays and do a cell assignment. This is one area of spatial biology that is really wide open with a lot of action, which is how do you identify which cell is which within a spatial microenvironment in a way that's very confidence lending. So I think, one way that the Plavridis lab has done this is using a semi-supervised method called Celesta. There are a number of AI foundation models that have been built that claim to be able to label cell type, and some of them are very good. And there's been, again, another comparison between these foundation models, finding that there are weaknesses in the different foundation models based on the rarity of the cell type that they're trying to call. But here, the Plavridis lab uses Celesta to call the cell types within their multiplex immunofluorescence assays. And then they build something called a co-locatome, in which they're looking at the co-location of cells within these sections. It's actually quite elegant in its simplicity, so I wanted to present it. So again, after Celesta cell type assignment, a quite easy co-location quotient is calculated, which is really a measure of how close different cells are in proximity to each other. Once you calculate that CLQ, take the data and permute the data by 500 times. So recreate these spatial sections over and over and over using different mixtures of the cells within one slide, creating a distribution of, this is a null distribution because these co-localizations don't actually exist. And then you test to see, does the real co-localization lie within that null distribution? If so, then that's not a real co-localization. And if it's outside of that null distribution, it is a real co-localization, which is an inferred some sort of interaction between these cells within this section. And this is really similar to the types of analysis that microscopists have done for a long time to look at the co-localization of proteins within a cell or within a tissue. So now we're applying that to cells. So for each of these co-localizations, again, inferring some sort of biological interaction between cells, You can ask if that co-localization quotient is maintained across either biological replicates or across different model systems. And what they ended up doing in this paper is looking at the CLQ value across the assembloids, so the in vitro system that they built, versus the human lung cancer sections that they're looking at, and found that there are a number of co-localizations that are important in the human context, in the human tissue context, that are not only spontaneously formed in these assembloids, but also evolve over time similarly when either the human tissue, the human tumors are treated with drug, or the assembloids are treated with drugs. So inferring here that we can actually learn a lot about spatial biology by carefully characterizing our in vitro systems that we're using. And all the data from the Cancer Systems Biology Consortium and the paper that I just talked through is available on the Cancer Complexity Knowledge Portal. This is a resource that has been built by Sage Bionetworks, and it contains data from 6+ Division of Cancer Biology programs. Another one of those Division of Cancer Biology programs in which the data is made available through the Cancer Complexity Knowledge Portal is the Cellular Cancer Biology Imaging Research Program. The goal of this program is to create microscopy and imaging techniques that span many scales, from the molecular all the way up to the organ scale. This program is interesting in that the purpose of it is to create technology solutions to answer specific cancer biology questions that are then pushed out into the community for wider use. One of the examples of the technology that has been supported by the Cellular Cancer Biology Imaging Research Program is a technology out of Johns Hopkins. This is a grant that's led by Denny Wirtz, but the CODA 3D image reconstruction technology was really first made by Dr. Ashley Kyman, who is a early stage investigator at Hopkins. And this imaging technology is meant to provide a three-dimensional picture of a tumor or any other tissue. What you're looking at here is a movie of about 400 individual 5-micron histological sections that are imaged at high resolution and then combined, which, by the way, is not an easy thing to do, to combine that many sections, register them. And then they use an AI model in order to recognize large anatomical structures within these large tissue sections. So what's cycling through here is their identification of blood vessels, islet cells. This is a piece of pancreas tissue. And this study is interesting in that what they really wanted to do was find out how many normal, quote unquote, normal human pancreas contain pre-malignant lesions. And in red, as this cycles through, you'll see Pannin lesions, which are pre-malignant lesions of the pancreas. And by doing this large scale visualization, what they find is that Pannins are actually continuous structures that tend to form around blood vessels. So that's an imaging technology that is being pushed out into the community through support from the CCBIR program. Another team of investigators within this program are at UT Southwestern. They're building technologies to help people use light sheet microscopy. They have an upcoming workshop here in September that is a hands-on workshop for a week, providing folks an idea of the types of questions that can be answered with light sheet microscopy, and then tutorials on how to build the systems that they have in their labs in your lab. These technologies that I've mentioned, many of them were initially supported by the NCI Innovative Molecular Analysis Technologies, or IMAT program. The IMAT program is NCI's venture into supporting only technologies. So there's no hypothesis that is required. In fact, you can't have a hypothesis in these grant applications. These are technology development, purely for technology development's sake. The next due date for the IMAT program is in early October. Partnering with these experimental technologies, we know that there needs to be development in the informatics and the analytical space. So the NCI also supports the informatics technology for cancer research program that I mentioned previously. There are 4 different types of ITCR awards that support all the way from early stage development of a computational method to software sustainment. So for example, the CBio portal, very well used, community loved platform, is sustained by one of the ITCR U24 grants. We've been looking at a lot of imaging here, and one of the things that the community really needs is ways to interact with imaging data. In an ITCR grant from Dana-Farber Cancer Institute, from Hans-Peter Pfister and Sandro Santagata, along with Peter Swerger, they've developed a imaging platform, imaging visualization platform called Minerva. And Minerva helps you to go beyond just having a static image in a paper to building stories that allow you to explain exactly what it is the finding is from this really interesting imaging technology or spatial biology technology. So for example, this is a picture from a Minerva story out of the Harvard Tissue Atlas. They had a melanoma precancer center in the Human Tumor Atlas Network program. And you can walk through and provide a narrative to the audience as to what they're looking at, why it's important, and put into context what is shown just in a static snapshot in any figure in a paper. The Minerva software is also being improved under this award to work with another technology developed at Harvard called Orion, which partners both histological imaging. You're looking at this H&E kind of within the bubble stain that shows you where tumor cells might be within a section, partnered with the highly multiplexed imaging, immunofluorescence imaging. So one thought here, one way to use this is to teach pathologists and people interested in pathology, how do you relate a histological section to the actual action that's happening within the tumor, the protein expression, maybe the spatial transcriptomics that we're all interested in. So this is another version of Minerva that's being supported by the ITCR program. And again, really important for us to be able to convey these spatial biology findings to the community. I'm going to end just with a couple slides on upcoming events. So as part of a combination of the Cancer Systems Biology Consortium and another program that we have at NCI that is supporting synthetic biology, we're really interested in understanding how we can create systems to test these spatial relationships that we're finding. So I talk a little bit about how we make assembloids or organoids in order to perform experiments that are validating some of our hypotheses, but can we actually engineer these interactions to learn more about the biology, or to engineer these interactions as some sort of therapeutic. So we're going to be having a virtual workshop series coming up here in mid-May through the end of May that will be followed by an innovation lab in order to dive more into how we can partner synthetic and systems biology. I don't probably have to tell this audience, but sharing spatial biology data is a huge challenge. These are giant data sets. This presentation was 200 megabytes just of beautiful imaging. So the NCI is really interested in hearing from the community how we can best support data sharing, especially in the spatial biology realm. So every year, NCI, the Office of Data Sharing, hosts a symposium to talk about best practices in data sharing. This year's symposium is at the end of September. Anyone who's interested can attend and help us to help you to share the data that's being generated. That's all I have, and I'm happy to take any questions. Go ahead and take my slides down.
Shannon, thank you so much, and thank you for persevering in a suboptimal venue for giving a lecture like this. We really appreciate it. I'm glad that you kind of touched on the data sharing elements of it, because I think that's so important especially for our Biomedical Data Science Innovation Lab participants this year, as they're going to be interested in where they can get their hands on some data. What are some of the kind of rules of the road? What are some of the things that people can do and could do now in order to start getting access to these data?
Yeah, well, first of all, I am more than happy to share these slides with anyone. And I did flash up my email address there, but it's just shannon.hughes@nih.gov. And within these slides, there are a lot of links to different data resources. Most of the data that I talked about is open access data. It's not controlled access data, especially the imaging data right now. So those types of data sharing, when you find it, and you have the capacity to download it and store it, it's pretty easy to do. But I think, some of these, there are other resources within the NCI. I talked about the genomic data commons. The NCI actually has a whole slew of data commons, the cancer research data commons, and that does include a data commons that hosts imaging data. More and more data commons, not just at NCI, but other places at NIH and also other places, other foundations and things that are hosting large data sets are encouraging people to work in the cloud with data versus downloading it themselves. So becoming more familiar with cloud platforms and different ways to interact with cloud data through Jupyter notebooks or what have you are also great ways to think about reusing the data that's out there. Because there's a huge amount of not just cancer genomics data, not just imaging data, but also flow cytometry data and other data types available.
Shannon, I'm glad you mentioned computing, because lots of people are interested in cloud computing. You mentioned there are a variety of different vendors and whatnot out there that provide those services. Does the NIH have any particular arrangements with any of the major vendors about cloud computing resources that directly link to any of these data sources?
Yeah, so yes, they do. And I think, so there are a couple answers to that question. So NIH, at the NIH level, has the Office of Data Sharing Strategy, or ODSS, and they have some resources for connecting folks to the big cloud data sharing platforms like AWS or Google, and providing resources through the NIH Strides program for people to start using data in that manner. NCI also has an independent program through the Cancer Research Data Commons, which will provide cloud credits for people to start using data within the cloud through things like the Belsera Cloud or ISB, Institute for Systems Biology. So you can apply to get these cloud credits from NCI so that you're not spending your own lab money. You're spending the NCI's money to kind of make the mistakes that you need to make first before you become proficient at interacting with data in the cloud.
I make a lot of mistakes, Shannon, so that would be a very expensive thing if I had to pay for them. So, speaking of paying for things, you mentioned, I think, with the Human Cancer Models Initiative, that the ATCC, I don't know who they are. Can you say a little bit more about who they are, what their role is in it? And you mentioned that you can buy certain models from them. How much would we expect to pay street value for one of these models?
Yeah. So ATCC, it's American type. I don't know the acronym, unfortunately, but it's a company that anyone who's done any sort of cell culture or worked with virus or others are very familiar with. It's a company that provides many different types of cell lines to the community, and NCI partnered with them because it is expensive to maintain and grow these types of advanced models like organoids. If you were working in a commercial company, the price on the HCMI models is somewhere around $2,500 to $3,000 per model. If you are in an academic setting or a nonprofit, it is significantly less, somewhere around, I think it's $500 to $750, which, again, these are models that, for the most part, are self-sustaining. You can use them many times to do the assays that you need to do, as long as you're culturing them in the right conditions.
So one of the things that's really interesting about these different models, some of the beautiful work that you showed us with the perturbed maps, and I was struck by just CRISPR, another really cool application of the CRISPR technology. I think it keeps going from strength to strength. But what about the computational requirements? What about some of the algorithmic things that what would you envision are kind of required to take some of that large data and really take it next level from a data science and informatics point of view?
Yeah, thank you for that question. So one of the things that I think some of these images mask, these beautiful images that we can produce, I don't want to say easily. I spent a lot of time sitting in front of a microscope, so certainly not going to claim that kind of thing is easy, but is the fact that we have to be really careful in the way that we analyze this data, because you can discover relationships that are not statistically robust by not really understanding how cell segmentation works, maybe not being very careful in how we're labeling our cells. I mentioned that understanding what cell types are being identified in these spatial biology assays is far from being a settled matter. And identifying cells by hand is a painful and very long process. So there are a number of people who are trying to build computational models that identify cells in a unbiased way or a semi supervised manner. But those are computational challenges that need to be overcome. There's a long standing challenge with just cell segmentation. And as we do, most of what I showed you, with the exception of the work from Johns Hopkins, was in a 2D environment, right? So we're looking at just a flat snapshot of cells, and it's difficult to segment those cells so that you're asking questions about individual single cells just within a tissue context. Now think about where we really want to go is we really want to understand how these cells are interacting in 3D. We know from work that I didn't have time to show today that some of these structures that we think of and pathologists think of when they're looking at patient samples as being individual cells that are kind of floating or somewhere near a tumor aren't really individual cells. They're actually fingers that are coming off of tumors. This was shown really elegantly by Peter Sorger's lab in colorectal cancer, that these cells that were traditionally thought of as being a sign of advanced cancer because they were potentially motile cells away from a tumor mass, actually are these really amazing and quite scary kind of fingers and tubules that are growing off of tumors and are not individual single cells at all. So how do we go from segmenting something in 2D to looking at the whole thing in 3D in a way that is computationally solid and savvy? So there's a huge amount of that's even processing. That doesn't even get us to how do we recognize repeatable neighborhoods that are predictive of patient outcome. So this is really a huge computational problem and a pretty exciting one for people who are motivated to try to solve it, I think.
I was struck by the comments about the tumor neighborhoods and the work that's going on in organoids and assembloids, because not only can you use those as a model system to look at the things you just described, like who your neighbors are, but it's not only in 2D, it's in 3D, and maybe it's even in 4D, in which case you've got a time element as well. And so you can look at not only, whose one's neighbors are, but the evolutionary kind of the whole development process of cancer in an assembloid, for example. Do you have any examples, or can you think of any examples where people have tried to do that?
Yeah, that's a great question. So there are a couple. So first, I'll answer that by saying that people are trying to infer that from snapshots. So for example, in melanoma, you can obtain a patient sample that has both full-blown cancer, so invasive melanoma, as well as precancerous lesions and normal skin, all in the same tissue. And so people are trying to infer kind of that progression of time just from samples like that. So that's one way to introduce that dimension of time. In an in vitro setting, there are also folks who are using advanced tissue engineering techniques to watch basically these organoids form, and these, in some cases, the right combination of cells can give you even the correct pathological structures that you see within tissue. And so there are people like Christina Curtis at Stanford and others who are doing that gastric cancer, for example. Dynamics, man, that's a challenge. That's a challenge experimentally. It's also a challenge computationally, right? To know how to handle that kind of dynamic data in the types of models that we want to build.
Oh, I think it's going to, already you're dealing with very large data sets, and 2D, you add a third dimension, that's going to make them even larger, and then add a fourth dimension of time, and all of a sudden these things just explode. And so it's really going to require, really sophisticated computational hardware, whether it's, on premises or in the cloud, to be able to process that data. But also it's going to require some really clever algorithmic stuff. And you mentioned cell sorting, for example, and trying to identify the cells. You can buy a machine that will do that, but clearly doing it computationally is going to be really helpful for us to be able to make inferences. How do you envision kind of the data science component of this kind of evolving in the next few years?
Yeah, so I know that Sean Hanlon was here this year as well to talk about the Human Tumor Atlas Network.
Yeah.
And one thing that we did last fall, and this is actually the second time we did it, is we held a data jamboree with the HTN data. And we had a super clever team who wanted to build an agenic model on top of our data portal, right? So you could come in and, ask just about anything you wanted and have it served up from you just from the HTN data. And I think that combining if we could be smart about the way we stand up data commons, and we can be smarter about the way that we label data, having consistent metadata across many projects is kind of it's a dream, but it's really difficult. We could move towards these types of agenic kind of conversations that we have with data portals to do the analysis that you want to do without having to be the expert in writing all of the code behind each one of the individual analysis. So I think that that's a huge opportunity. It relies on having data we can trust. And so things like the Genomic Data Commons, the HCMI program, where the analysis was all done on the same experimental pipelines and then processed through the same analytical pipelines, helps to gain more trust in the types of data that go into those types of chatbots and things that we hope will help us make sense of this huge amount of data we're generating.
Well, Shannon Hughes from the National Cancer Institute, thank you so very much for sharing this with us. I mean, these are just such wonderful resources, and I'm really anxious to see some of our Biomedical Data Science Innovation Lab participants taking advantage of these, looking at them, doing a little, some analyses of them to kind of get a sense of how they can turn these into projects that they'll hopefully pursue with data science experts, and hopefully identify some possible grant application opportunities. And obviously with your help, we'll be able to do that. So thank you so much for sharing this with us. We'll look forward to seeing you again when we're having our micro labs and having our in-person event in Nashville. I want to just thank everybody who has tuned in throughout the year since we began these back in October with all of our Foundations of Biomedical Data Science seminar series. So thank you so much for those of you loyal visitors who keep watching. This is our last one for this year. We'll plan to start things back up again in the fall. I really want to thank Gina Kamdar and Hannah Kears for their dedication to this and all the stuff they do behind the scenes. And we are just really looking forward to doing another Biomedical Data Science Innovation Lab. This will be, we've touched on so many different things, but we're really excited about this year's activity, and we're looking forward to getting everybody together in person. And Shannon will be among those folks who join us. So thanks, everyone. Have a wonderful summer, and we'll see you in the fall. Bye now.